before the first plenary lecture, I'd like to make several short technical announcements. The first one is that all talks will be registered, and then the links will be announced. Then we would like to ask all speakers to send us the slides of their presentations in advance, better the day before, because sometimes there are some technical problems, as for example now, and sometimes speakers cannot show the presentation. So in this case, the organizers will do it, but we need the slides, so please do that. The next point is that we open the connection 30 minutes before each session, so we would like to ask all speakers to connect in advance to check the connection and share screen option. And finally, we will try to respect the time schedule. So please also make sure that your talks are start and finish on time. So thank you very much. And now it's a pleasure for me to announce our first plenary lecture. Professor Bocharov from Marchuk Institute of Numerical Mathematics will talk us about mathematical immunology, emerging challenges, and novel approaches. Please, Gennady, we are listening to you.
Thank you very much for the introduction, and I would like first to thank the organizers of this conference and Professor Vitaly Volpert for the opportunity to present our recent studies in mathematical immunology. Next slide, please. So the structure of my talk can be specified as follows. First, I will make some introductory remarks about the immune system. Then how the field of then I will describe how the field of mathematical immunology emerged. Then we will consider two most essential, in my view, aspects of modeling the immune responses, and these are related to the regulation and the structure and the structural aspects of the immune system functioning. Oh, please, next slide. So the I would like to start my talk with the. I would like to start my talk with the quotation of what was considered as a credo of Professor Rolf Zinkernagel, who got the Nobel Prize in physiology or medicine in 1996. So what is the primary function of the immune system? The immune system is primarily about host survival of infection, and for this we also need to know the biology of the system. Next slide, please. And what are the basic components of the protection against pathogens, which represent the function of the immune system. So on the left figure, on the left side, you can see the basic scheme or basic representation of the structure of the pathogen, which expresses on its surface a number of various antigens. These antigens are recognized by immune cells, B cells or T cells. Also the recognition of the antigens by T cells is more demanding in terms of the cooperation with antigen presenting cells. And then those lymphocytes that received a strong stimulation via a B cell receptor in this particular case are selected and expand to produce a progeny of B cells and plasma cells, which secrete antigen-specific antibodies, and this, I mean, provide the protection against the invading, I mean, pathogen. So, and in terms of the I mean, population dynamics of the immune responses and the, I mean, pathogen load, one can um, I mean, present the, I mean, following scheme of the phenotypically different forms of the infection, I mean, dynamics. It could be a subclinical infection, it could be an acute infection, it could be a little infection, or it could be a chronic infection. And understanding the, I mean, factors that, I mean, underlie the emergence of, I mean, different forms of infection is actually the, I mean, subject of both classical, clinical, and mathematical immunology. Next slide, please. So what are the difficulties associated with the analysis of the immune system functioning and in, I mean, particular with the development of the mathematical description of the immune processes? The major problem is due to the complexity of the immune processes. The, I mean, complexity can be, I mean, expressed in, I mean, different, I mean, categories. First, as one can see on this slide, it's a multi-scale, I mean, phenomenon when we consider the, I mean, interaction of the pathogen with the host, which spans many orders of, I mean, magnitude of the spatial scale and the, I mean, temporal scales. Next slide, please. And the other, other aspect is the multi-physics nature of the immune system, I mean, processes. So one need to, in order to, I mean, formulate a comprehensive description of the immune, I mean, responses to, I mean, pathogens, one need to consider in the models to, of course, to I mean, a various degree of detail the physical processes, the chemical process, and the biochemical process. And the fundamental challenge to applied mathematicians is to understand immunology in new ways by using models, computational techniques, and, I mean, algorithms. Next slide, please. Since the beginning of the 21st, I mean, century, the field of experimental and clinical immunology has been, I mean, characterized by I mean, penetration and the application of, I mean, novel high throughput, I mean, measurement, I mean, technologies, which led to the generation of enormous amount of, I mean, data characterizing the development of the processes at the I mean, genetic level, at the cell population level, at the single organs, or the whole I mean, system, or the whole, I mean, organism level. So this one could just name few of them, like transcriptomic, flow cytometry, I mean, PCR, I mean, analysis, I mean, quantitative single cell, I mean, RNA, I mean, analysis, and the, I mean, imaging, confocal microscopy, etc. And of course, the generated data set, they span I mean, different types of I mean, they produce, I mean, different types of data, the cell population data, the, I mean, categorical data, I mean, etc. And one need to develop appropriate I mean, mathematical tools that would enable a researcher to assimilate the, I mean, data, to describe them, and to interpret in a, I mean, in an immunologically I mean, consistent and informative way. Next slide, please. And the complexity of the immune system called for the application of the systems approaches to, I mean, immunology. By definition, systems approach requires the implementation of, I mean, mathematical models and, I mean, mathematical techniques in the analysis of the immune system. And the systems approach implies an integration of data and process across a number of levels of the immune system organization, as shown in this cartoon. And this is a genomic level, cellular level, physiological level, and the resolution and the level of complexity, of course, differs enormously. And the systems approach to immunology is based on the triad: the experiments and the experimental data, the theoretical concepts that are used to interpret the experimental data, and the mathematical models which are to be developed in order to assimilate, describe, and analyze the data. Next slide, please. So let's consider first the theoretical foundations of immunology. One can distinguish the following well-established concepts of immune regulation. So the first one is the well-accepted clonal selection theory, which was proposed by Nobel laureate Francis Burnett. The second one was the network type immune regulation, which the theory didn't get support in experimental immunology, and finally was rejected. The spatial temporal view of the rules for immune responses regulation. It was proposed by Rolf Zinkernagel in his CREDO paper. The balance of growth and differentiation concept proposed by Zvi Grossman and Bill Paul from NIH. In some of the implementation in mathematical models of some of these concepts, in particular the clonal selection theory and the balance of growth and differentiation, will be considered in the next part of this presentation. Next slide, please. So, what are the scientific questions that have to be addressed in if one is going to follow a systems approach? So the immune system functioning can be structured and emerges from the interaction of these following three categories of its properties. So the dynamical properties characterized by the data on the temporal evolution of the cell populations and the pathogen load and the physiological characteristics of the host. Then second is the regulation of the immune responses, and this is particularly important because many effects of the same molecule or the same population of cells are pleiotropic. So they have the ranges in which they positively affect the response of the system, or the negatively affect the response of the system, as shown in this middle figure. And finally, one also need to consider the structure of the immune processes in the whole organism and in the lymphoid organs. And as far as the mathematical modeling in immunology is concerned, most of the model have been considered so far the dynamical aspects of the immune responses, which with a much less attention given to the regulation and the structure of the immune responses. Next slide, please. So we will move on to consider the roots of mathematical immunology, and we will highlight some differences between the initial models of the immune responses to pathogens, which are related to the way how the regulation of the immune responses was considered. Next slide, please. So I would like to refer to the viewpoint of the outcome of any interaction of the pathogen with the host organism as a numbers game, or as the race between the ability of the pathogen to spread in the host and the ability of the immune system to mount a reaction against the invading pathogen. And the quantitative view and the conceptual view that was shared by immunologists and mathematicians, in particular Rolf Zinkernagel, professor and academician Guri Marchuk. It's that the outcome of infection results from the numbers game between infectious agents and the immune system. And the mathematical models, and one of them is just shown here. It's the so-called basic model of immune response. Allows to quantify the parameters of this numbers game, and hence serve as a predictive tool for the analysis and prediction of the outcome of the pathogen-host interaction. Next slide, please. So, what is the role of mathematical models? Immunologists expect that the mathematical models, of course, should be able to quantitatively and qualitatively describe characteristics of the process dynamic. They should help in interpretation of experimental observations. And in particular, it is very important that models should be able to generate testable predictions, so suggestions of new experiments, or highlight the hidden effects. But the model formulation requires continuous interaction with experimentalists or clinicians, and the calibration of the mathematical models requires a solid input from experimental and clinical research in terms of the precise and comprehensive data sets. The other aspect is that certain components of the mathematical models are based on the views which are not completely biologically understood, and this leads to the formulation of a family of plausible models, and the need to select between them the most informative and the most optimal one. Next slide, please. So the two founders of mathematical immunology, who established scientific schools long-standing scientific schools in this area, are Professor George Bell from the Los Alamos laboratory, and Professor Marchuk from the Academy of Sciences, and so they develop to a certain extent similar mathematical models of the immune response to replicating antigen. The only difference was how the regulation, what was the detail of the consideration of the regulation of the immune responses. And in the model of immune response to replicating antigen proposed by George Bell, it was considered the predator type of the regulation of the immune responses. So the pathogen acted, produced the activation signal for the cells of the immune system. They expanded, and they led to the reduction of the pathogen load, a pure predator-prey type concept. And in the model proposed by Professor Marchuk, it was a model of infectious disease. It was the damage to infected organ that was additionally considered. And this, damage to the infected organ, either by the pathogen or by functioning of the immune system, led to the emergence of the suppressive loop in the regulation of the immune responses. So it was, there were two regulatory loop which were competing in the induction of the immune in the regulation of the immune responses. So the predator-prey type and the suppression of immune system by damage of the organ. Next slide, please. However, the data which characterize the emergence of the acute versus chronic infection produced by the Institute of Experimental Immunology in Zurich, led by Professor Zinkernagel. They found a fundamental property of associated with the development of the chronic infection. It was a so-called exhaustion of pathogen specific excuse me, immune cells. And this slide shows the two phenotypically different types of the infection dynamics. The green line and the black line characterize the acute infection with the development of the antigen-specific immune response and the persistence of the lymphocytes in the form of the memory. And the lower panel specifies the qualitative view as well as the data of an opposite phenotype, which is characterized by initially an induction of the CTL response and then complete exhaustion of the pathogen-specific immune cells. And in order to describe this type of behavior, and in particular the quantitative data, part of those shown, on the left part of this, figure, one had to incorporate two additional regulation loops in the model of the immune response. Next slide, please. And these were associated with the consideration of the apoptosis and the energy induction. The question is how this could be parameterized in the mathematical model. Next slide, please. One can see here the set of four nonlinear delay differential equations which describes the immune response to LCMV infection. And in addition to the standard components of the models, like the replication of the pathogen, elimination of the pathogen, the homeostasis of the immune system, the clonal expansion, one needs to consider the induction of the energy and the antigen and the activation induced cell death by apoptosis, both marked in red and blue. And this is actually implies the incorporation of an integral negative feedback regulation of the immune response in LCMV infection. Now, next slide, please. I would like to make a more specific focus on how the regulatory loops or the regulatory motifs can be specified in the analysis of the immune responses. And in particular, I will refer to a study made together with experimental colleagues from the laboratory of immunology of William Paul. It concerns the regulation of the clonal lymphocyte dynamics to non-replicating antigen. Next slide, please. And the particular phenomenon that was addressed was the following. On the left panel of this slide, one can see the population dynamics of the antigen-specific CD4 T cells when for a low number of precursor cells and for a high number of precursor cells on top and the bottom of the figure. And the remarkable feature that was found was the following. If one increases the number of precursor cells by a factor of, for example, 100, then the...
excuse me, Gennady, we don't see the figure, we see the title of the paper. Is it a correct slide?
No, next, it should be next slide.
Thanks.
Thank you. Next slide. Yeah. This is the one. Yes, and then one can see here, exactly the phenomenon. So that the factor of expansion depends not on the increase in the factor of expansion of antigen-specific cells produced by clonal proliferation depends on the square root of the factor of increase of the initial precursor number. And this is a remarkable phenomenon. So it's not proportional to the extent of increase of the precursor cell number, but to a square root of the increase of the factor of increase of the precursor cell number. And the question is how this kind of phenomena could be described with a mathematical model. Next slide, please. And for this, we had enormous data sets, which characterize the time course of cell population dynamics for different numbers of initial antigen-specific cell numbers. We had the data on the factor of expansion. We had the data about the division H structure produced by CFSE dilution. This is a fluorescent dye. And we had the cell cyclic, fraction data produced by the incorporation by another, marker, BRDU, as shown in this figure. Next slide, please. And the process was extremely, detailed in terms of the different lines of mice, different ways of adoptive cell transfer and immunization, the analysis of the in vivo proliferation and division rate, enumeration of cells using real-time, PCR. And next slide, please. And the analysis of the data set suggested us to conclude that this factor of expansion dependence on the precursor number implies some type of local feedback control that need to be identified using mathematical modeling approach. Next slide, please. And we have, these are just few examples which show the dependence shown on this in the right, bottom part of the figure, that the logarithm of factor of expansion is minus 1/2 of the logarithm of the precursor number. So exactly the phenomenon that we ought to explain in terms of the regulation mechanism. Next slide, please. And so this slide just shows the various types of the data that we had to incorporate in the process of the model formulation and identification. Next slide, please. For this, we used the maximum likelihood approach. We used the information theoretic ranking criteria. Next slide, please. And finally, we had to identify the strengths and the mode of the impact of the cell populations that regulate their function, functioning of each cell population subsets. So to this end, we use the concept of feedback regulated balance of growth and differentiation. So at any given time, the growing cell clone consists of the proliferating and differentiating T cells, and X1 and X2 are the proliferating cells which differ in their maturity, and they differentiate into Z1, Z2 cells, which don't have the ability to proliferate, but can exert the effector function, and in particular provide some feedback on the process of the maturation and differentiation of the dividing cells and the less mature cells. Next slide, please. And we have in figure A, we had the general scheme of all possible interactions and the regulation in this system, which was alpha specifying the constant F and F specifying the functional dependencies of the intensity of the regulation on the abundance of X1, X2, Z1, and Z2. And finally, we arrived by combination of model calibration, the information theoretic criteria-based ranking of the model to a final structure. Next slide, please, which is shown in this here in figure B. And in order to assimilate the amount of data that we had, the basic model consisting of considering four different cell populations has to be expanded to incorporate the division structure of cells in order to be able to be considered to consider the BRDU data set. Next slide, please. and also the CFSE data set. And this led to enormous expansion of the dimensionality of the model. But finally, this led us to a comprehensive description of an assimilation of all data sets. Next slide, please. So the cell population dynamics, the BADU incorporation dilution data, CFSE dilution data, as shown in this figure. And finally, we led to the, next slide, please. And finally, we identified all the parameters of the best fit model and the optimal model in terms of the Akaike, criterion with the 95, percent confidence intervals as shown in this table. Next slide, please. And these are just a few examples of how good the model describes the data on BRDU labeling and CFSU dilution data. So different colors refer to data, to experimental data, the model behavior, and one can see that it's really consistent with the data, with experimental data. Next slide, please. So this is the final motive underlying the feedback regulation of the CD40 cell population dynamics, and this characterizes the design principle of the regulatory feedback in the immune system and the pattern of interactions between cell population. Next slide, please. So finally, the model allows us to refine the quantitative relationship between the factor of expansion and the precursor number, and the dependencies that were parameterized with the model are just shown here at day 7. How the factor of expansion depends on the precursor number, and how the size of the clonal expansion depends on the precursor number. Next slide, please. In particular, I will skip the next part because I need to consider the analysis of the structural properties of the immune system. But one should also mention the more complex mathematical model of antiviral immune response proposed by a joint work of immunologists and mathematicians. So Professor Petrov and Professor Marchuk, which was based on the concept of MHC restricted antigen recognition principle. And this model was, we used, recently. Next slide, please. It shows the structure of the model. It's a set of 10 delay differential equations with 5 delays. Next slide, please. And it was used to analyze the mode of interaction of antibodies and CTLs in protection against infection. And we show that, according to this model, these two primary control factors of virus infection are cooperating multiplicatively rather than, additively. This implies that in order to achieve a certain level of protection, one can either increase single arm by certain factor, say by a factor of n, or it can only increase each component of the immune response by a factor of square root of n, as shown on the slide, on the 2 slides below. Please go, please skip the next slide and the next slide. Yes, you see this green figure shows that the mode of cooperation of the antibody-specific response and CTL response in protection against infection is, characterized by a multiplicative nature. Next slide, please. And this allows us to make predictions about the requirements for protection against influenza infection, HBV infection, or simian human immunodeficiency virus infection, as shown in these slides. But now I would like to go to the last part of my talk. It concerns the structure of the immune system. The immune system is very complex. It's a body-wide network of the lymphoid organs, of the primary and secondary lymphoid organs, and has about 500 to 1,000 lymph nodes, as shown in this figure. Next slide. And one can use the information about the network structure of the lymphatic system in order to generate the computational models and the graph models of the human lymphatic system. And here I would like to go to some historical data. So the first oriented graph of a simple network model of the human lymphatic system was proposed very long ago by scientists from Texas University. See, here one can see the structure and the complexity of this graph network. It has 29 nodes and 29 edges. Next slide, please. The second one was the recent graph model of the human lymphatic system proposed by our colleague from the Moscow State University, by Mukhin and Mozokhina. And the version 3 graph. Next slide, please. Next slide, please. Next slide, please. This is a ready graph. Next slide, please, is a Mukhin and Mozokhina graph. And next slide, please. This is a graph that we know. Just one before, graph that of the human lymphatic system, which is based on the anatomical data, which one can find at this website. Indicated here is a plastic boy, and the graph and the oriented graph that was generated considers about 1,000 vertices and about 1,000 edges. Next slide, please. And finally, we move to the description of the computational lymph node. The first paradigmatic computational model of the lymph node is shown here. It was published quite a while ago. Next version was the model proposed about few years ago. It is based on the exact imaging data and the analysis of the topological processes of the fibroblastic reticular cell network. Next slide, please. Next slide, please. Here's the global view of the computational lymph node model of the generated some time ago. And recently. Next time, please. And recently, we generated version 3. So it's a 3D model incorporating the structure, the topological properties of the conduit system and the blood vascular networks. And now I would like next slide, please. Next slide, please. Now we have enormous data generated by Jimmy Moore team from Imperial College, and the data produced by the group of Volpert, Ludovic, which characterize which quantify the whole lymph node vasculature, vascular structure and the conduit and FRC networks. And this calls for the need for the opportunity to develop a computational model of lymph node version 4. Finally, next slide, please. What are the research directions that we expect to be followed? So we need to be able to embed the cell and cytokine and lymphocyte dynamic and lymph dynamic models into the spatial context of the lymphoid organs and the peripheral tissues. And to this end, we need to we also need to develop and better understand the multi-scale and multi-physic regulation of the structured immune cell population dynamics, and to develop tools which would allow us to assimilate various types of data which characterize the cellular function, cellular dynamics, the imaging, and etc. And this next slide, please. So these are the acknowledgements that I would like to express to my colleagues from St. Gallen, from Barcelona, from Bethesda, and from the Lomonosov State University, and the funding which was used to conduct the presented studies. Next slide, please. Thank you very much for your attention.
Thank you very much for your very interesting presentation, this overview of mathematical modeling in immunology. So we have several minutes for questions, and our listeners can ask questions either by using chat or raising hand, and then you will be allowed to switch on your microphones. So while waiting for other possible questions, I would like to ask the question which is often asked when we discuss mathematical modeling in biology, and in particular, of course, it is very important in modeling infectious diseases. So this all this so complex immune response to viral diseases, and it can be very different in different patients. So what do you think, whether it is possible to get enough data to model patient-specific response to viral infection and to optimize antiviral treatment according to each particular patient?
Mm-hmm.
So the question about can we get data from the experiments, from clinical measurements, enough data to model it in a patient-specific way?
I mean, thank you very much for this, fundamental question. I would like to rely my answer on the experience that we have. So basically, we were, maybe the patient-specific, prediction and the treatment is still, ahead of us. But at least what we can do, we can analyze the different phenotypes of infections. For example, we have an experience of how mathematical models can assist in understanding what would be effect of using certain antibodies that inhibit immune control checkpoints and are broadly used in cancer treatment. For example, the antibodies to PD-1 receptor or to PD-1 ligand. And for example, in HIV infection, this is not a clinically accepted method, but because the immune cells in a chronic or in HIV infection are shown to be in exhausted state, so it seems to be promising to use this blockade or the inhibition of immune checkpoints in order to restore the functional properties of the T cells and the B cells. And one can consider different phenotypes of HIV infection, which are well accepted in clinical immunology, like this: rapid progressors, slow progressors, non-progressors, elite controllers. And depending on the level, on the combination of the level of the amount, on the level of the neutralizing antibodies and CTL and the viral load and the extent of immunodeficiency, one can predict with mathematical model what would be, if any, benefit in terms of the reduction of the antiviral load or of the viral load and antiviral immune responses in quantitative terms. Or so if one applies a combination of highly active antiretroviral treatment plus inhibition of immune checkpoint of immune control checkpoints, antibodies, what would be the effect for different groups of the patients? This is the answer I can base on my personal experience with my colleagues.
Thank you very much for this response, and of course there are many open questions, and I hope we will continue our fruitful discussions in this workshop and outside. Mm-hmm. So yes, thank you again. Thank you again for this very important presentation, and we are going to move to our next speaker, Professor Moore. Try please to share your screen if you can, and then we will start your presentation. Yes, we see your screen.
Okay, good morning, everyone. And thank you, Gennady. That was a fantastic talk. I really enjoyed that. It's clear. You make a very good case for the complexity of the immune system. And I think that's very important for the audience to realize that, this is not not the most simple system in the world to take on with either experiments or mathematical modeling, and really, really elegant work. So congratulations for what you've done and what you contributed to the field. So I'm going to that also serves as a great introduction to what I'm going to talk about. So I will sort of speed through maybe the first few slides. I think most of us have some idea about the lymphatic system, and it's important in immunology. And just wanted to highlight a few numbers here. Typically you think about flow through the body, you think of blood flow, with cardiac output's about 5 liters a minute. Lymph flow is about 5 liters per day. Pressures are much lower in the lymphatic system. Diameters are smaller. Reynolds number is smaller. So it's a low flow, low pressure system, but absolutely crucial in terms of delivering that information that Professor Bocharov talked about into the lymph nodes, so that all of those processes that he showed in the latter part of his talk can happen. So we should not underestimate the complexity and the importance of the transport process as well. So, we think of this as information delivery, so which is facilitated by fluid flow in the lymphatic system in its various parts, not only the lymphatic vessel, but also within the lymph node. So if you think about, a vaccine or some kind of infection coming into the body, then that information needs to be gathered by initial lymphatics, typically, which exist in different densities in different tissues. And then there's an active pumping of each individual lymphatic vessel. For most tissues, the lymphatic vessels would be easily under a millimeter in diameter. We typically, when we're looking at pumping lymphatics, they're around 100 microns in diameter, or about the diameter of a human hair. And they do their own pumping, so they're small little check valves, and they squeeze and they pump the information to the lymph node, which could be several centimeters away from where the infection has occurred. And without that fluid flow, you would just not have that information delivered into the lymph node without in any reasonable amount of time. And then even within the lymph node, which is what I'm going to focus on more today, the fluid flow of the lymph and actually the blood through the lymph node is absolutely crucial in mounting an immune response. So this is what it looks like when antigen comes into a lymph node. These are not my results. This is from Mike Carroll's lab at Harvard. And so there you can see there's a small antigen coming in. There's a large antigen coming in. This is actually time in minutes here. And you can see really within just a few minutes of injection in the foot pad of the mouse, you start to see these antigens being delivered in very different ways and into different parts of the lymph node. The small antigen goes down the conduits that Professor Bocharov referred to, and these are the B cells that he referred to as well. So the blue B cells are trained to recognize the antigen. The white B cells are naive in that sense, but you can see they're all communicating with each other, and they're walking around and responding to the delivery of that antigen into the lymph node. And then the larger antigen comes in a bit later and diffuses down different structures in the lymph node. So the transport processes in the lymph node are really hugely interesting and a bit mysterious. We know of the importance of the conduits. We know that there is a fairly tight parenchyma in the lymph node, and presumably that's where the larger antigen is going in. It can also be ushered in by cells that line the covering of the lymph node here, so those cells will capture some of the antigen and just walk it in themselves. So the questions I'd like to address today are summarized here. So if we think about, there's this information that comes in through the afferent vessels in the lymph node. That fluid will typically get distributed around the subcapsular sinus, which is a relatively open space for fluid flow around the outside of the lymph node. Some of that will come into the B cell follicles, as you saw in the previous slide, and then some will come in through the interfollicular zone into the T cell cortex here, and then of course there's important communication between the T cells and the B cells at the borders of these B cell follicles, and absolutely fascinating processes. And then that fluid is eventually going to sort of be gathered by the medullary sinus and go out the efferent lymphatic vessel. Now, the thing that's not shown in here, which I will come back to later, is the structures of the blood vessels inside the lymph node. And actually, Professor Bocharov already referred to that as well. So we'll come back to that towards the end of the talk. But first, I would like to talk about some recent work that just got published from my lab, looking into the dynamics of lymph node swelling. And really, the lymph node swelling is very, very common. We've all experienced it when we get ill, having our lymph nodes swell up temporarily. And really, we sort of started thinking about this from a fundamental point of view. Why do lymph nodes swell? What purpose does it serve to have a lymph node swell when you get ill? So I'm going to address that first, and then we'll come back to the second question later. So throughout both parts of the talk, I'm going to be referring to a mouse model that we have used. And I think, of course, you have to, just like a numerical computational model, mathematical model of immune function, you have to be careful how you use experimental models of immune function. And, these mice provide somewhat of a basis to understand what goes on in a human. But I think really what we're after is, sort of trying to combine the experiments with the modeling and really try and understand, first of all, what's going on in the mouse, and then understand better how we can really apply that to the human. But we use an injection into the foot of the mouse, and then we look at what happens in the nearest draining lymph node, which is just under the surface. They're only about 1 millimeter in diameter, but you can find them with a bit of training. So that's what they look like. And then for the latter part of the talk, I'll go back to a series of flow measurements that we did where we pumped fluid through the lymph node and measured the resistance, hydraulic resistance of the lymph node. So I'll come back to that later. But first, let's look at the swelling. So when you inject toll-like receptor agonists into the foot pad of the mouse, and these are sort of remnants of bacterial membranes that are used to kick up the immune response. It's a common way of studying immune response, but they've also been used in some vaccines as adjuvants. So if you just inject PBS into the foot of the mouse, you'll see not too many changes. This is a serial scan through a typical lymph node in that case. And you'll see not much change in the lymph node, but when you add the toll-like receptor agonist here, you'll see that the lymph node grows by quite a bit. And if we weigh those lymph nodes, we see that they're about 3 or 4 times heavier after 24 hours, which is quite fast. If you think about the typical time scales of tissue remodeling and adaptation, for something to change its weight in 24 hours to this degree is quite unusual. And if you look at the volumes, you see similar numbers. Again, these structures are growing by large amounts within a very short time. Certainly some of that is a shift in fluid flow. I'll come back to that later. But there's also a massive increase in cell numbers. And that's what we want to focus on in the first part of what I'm going to tell you about today. So this is an agent-based model. And as I said, this is just accepted for publication. And if you'd like a copy of the paper, I'm happy to send that to you. But we've got our typical sort of view, similar to what I showed you before, the lymph node of stuff coming in, stuff going out, and then stuff accumulating in the middle in kind of a traditional conservation of mass sense. So we've got our grid for our agent-based model here. We assume that most of the T cells that come in are coming in from the blood vessels, namely the high endothelial venules, across which most of the T cells in your lymph nodes have arrived. About 10% of the T cells in your lymph nodes will have come in from the afferent lymphatics. And then there are some sort of exit points here where the cells can migrate and leave the lymph node. And then so essentially we set up a massive grid here. We have our lymph node in the middle of it. And then as the lymph node swells, it just takes over more and more of these grid points here. And that expansion is a sigmoidal function of the T cell numbers. It's actually volume, not area, that expands. So, and then within the lymph node, within the grid, we can fit at most 2 T cells. There's a dendritic cell here, which is a bit bigger. They can interact with each other. These T cells can move about sort of randomly and communicate with each other and pass information from one to the other. And then if they get near to the exit, then they can exit as well. And their equations, I'm not going to go into the details today. It would take up the entire day to explain all the equations in the model. But it's you know, there's some conditions on whether or not the T cell can exit. Let's just say that depends on SPR1 and other things as well. So so there you go. I'll show you how the thing behaves under baseline conditions. So this is sort of what it looks like in the computational framework with the different cell types. This is about a day and a half after antigen presenting dendritic cells have come into the node. So we increase that up over the space of a couple of days and then let it die down over the space of a couple of days. And what you'll see is that the total T cell number goes up by quite a bit and then sort of tails off as time goes on. Effector T cells come up later on, and then activated T cells, cognate CD4 positive, CD8 positive T cells, memory T cells. You can see how these different populations come up dynamically over time and then sort of die away. And remember, though, these are the populations within the lymph node. In some cases, well, in most cases, as these cells are dying away, they're actually going out into the body, out the efferent lymphatic vessel to distribute that memory and the adaptive immune process, important part of the adaptive immune process. And you can see the egress rate here comes up over the first few days, and then really, you know, kind of remains pretty high after the antigen presenting D cells are no longer even coming in. So one thing we wanted to look at is how the swelling affects these T cell numbers. So sorry, I'm going to go back here and just remind you of the dynamics of these different processes. And really, we wanted to look at how this activated T cell number depends on how much you allow the volume to swell. So in these baseline results, it's swelling by about 2 times at most. So it can only get twice as big in terms of volume, and that's a parameter we can control. So we can allow it not to expand at all. So that would be a maximum volume of 1.0 normalized to the baseline volume. And then it can at most get about 3 times or 2.8 times its baseline volume. And you can see how the total number of activated T cells you know, goes up by about a factor of 2 if you allowed the lymph node to swell. And you know, some of the other cell types are not affected very much, and some are slightly decreased. And but if you know, overall, this is a pretty good number here, and pretty good indication that the reason that lymph nodes swell is so that more and more T cells can learn about the infection, and then they can pass that information on to their neighbors. And if there's more space to do that, then you can do that more effectively. So, and you can see how the T cell entry rate is governed by the amount by which you allow the lymph node to swell. So, so I'd like to go on now to the second question, which is when an infection comes in, how is that information delivery process, which again is somewhat determined by the flow patterns, the lymph flow patterns within the lymph node. How is that delivery process changed by the swelling? So here is a rough illustration of our flow schematic inside the lymph node. So the lymph comes in, it can go in the subcapsular sinus and then through the B cell follicles, T cell cortex, and the medulla, or there are actually direct connections from the subcapsular sinus to the medulla. And so there's a possibility that some flow will go that way as well. So that's what the subcapsular sinus looks like. As I mentioned earlier, it's a relatively open region for fluid flow to go through, kind of like parallel plate flow that we teach our undergraduate fluid mechanics students about. And then you can see how that compares to the relatively densely packed parenchyma and the T cells and the B cell follicles. T cell region and B cell follicles would look both about this dense in terms of the porous medium. The medullary sinus is a less densely packed porous medium, and partially for that reason, actually most of the flow is going to go around the outside. So this is sort of the path of least resistance. And again, this is under baseline conditions when there's no immune reaction going on. You can see the relatively high velocities of the fluid going around the lymph node and out the medullary sinus here, compared to the much lower velocities, about 3 orders of magnitude velocities. I'll point out these are in microns per minute. These are in millimeters per minute in terms of the color coding on this side versus this side. So only about 10% of the flow is going into the middle part of the lymph node where the B cells and the T cells reside. So we think this affects the delivery of antigens in suspension, and we're doing some looking at the way that the delivery down the conduits, as you saw in the movie in the first part of my talk, how that might be affected by the flow or not. And so we'll be talking about that in another conference in the future, hopefully. So going back to what I showed you earlier, so we take in these experiments with the mice, we can take those lymph nodes out and cannulate them, and these are exquisite little experiments. These lymph node, the afferent and efferent lymphatic vessels that connect those lymph nodes are only about 50 to 70 microns in diameter, and these are tiny little vessels, and to cannulate them is a real struggle, but my experimental colleagues can do this. And so we measure the flow across them, which is also very difficult. It's in sort of the order of microliters per hour. And then we also measure the pressure difference across them, which might only be 1 or 2 centimeters of water pressure difference. But we can do that, and we can get an idea of the overall flow resistance of these lymph nodes, keeping in mind they're only about 1 millimeter in diameter. So it's not really realistic to try and measure the flow velocities within the lymph node in this experimental setting, but we can get an idea of the overall flow resistance of the lymph node. You can see in the baseline case, the flow resistance is fairly small. In the activated case, so this is where they've received that the LPS and the poly-IC, so the activators, immune system activators that I talked about earlier. And you can see the flow resistance of overall of the lymph node has gone up a lot in 24 hours, but actually after 72 hours, it's come back down to its baseline state. And if you remember what I showed you earlier, the lymph node has already swollen up by this time here at 24 hours after the injection, but its volume actually remains high, even though the flow resistance has come down now. And why that is, we don't really know yet. We have some ideas that I'll come back to later. But really what we wanted to answer in this part of the experience is, all right, well, the overall flow resistance goes up, but what part of the lymph node changed in order to create that roughly order of magnitude increase in flow resistance? So here's the healthy state and the inoculated state and the flow resistance, same plot I just showed you earlier. And then we went back to the computational model and started changing it to see what would change the overall flow resistance of the model. And so we changed the permeability, the hydraulic conductivity of the T cell cortex. But remember, the only 10% of the flow is going in there under baseline conditions. So you can change that all you want, and it won't affect the overall resistance to flow of the lymph node. We can't, so that's not it. So the next thing we tried to do was change the height of the subcapsular sinus. We increased it, we decreased it, and it didn't really make a big difference. So here's the change in flow resistance when you cut the height of the subcapsular sinus in half. So not a big effect there. Certainly not big enough to explain what we saw experimentally. So what about the medullary sinus? Remember, there is this direct connection between the subcapsular sinus and the medullary sinus. And sure enough, when we change the hydraulic conductivity, by the medullary sinus from, basically change it by an order of magnitude relative to the T cell cortex, then actually we can get that increase in overall lymph node flow resistance that we saw experimentally. So we think there's something going on in this connection between the subcapsular sinus and the medullary sinus, some sort of cell type, and there would not have to be very many of them, that shuts down the flow around this path of least resistance so that more of the fluid can get shunted into the middle. And that's actually what we saw with our computational model when we increase the when we decrease the hydraulic conductivity of the medullary sinus, then sure enough, the flow into the T cell region goes up by about a factor of 3. And so, under baseline conditions, 90% of the flow will go around the edge. But when you have an immune reaction, more and more of that fluid, incoming fluid, will get shunted into the region where the T cells and the B cells are. And this is one of the figures that Professor Bocharov showed. This is our attempt, at least, to image the blood vessels inside the lymph node. And this is very difficult to do because it's truly multi-scale imaging. So the blood vessels that come in, so the artery and the vein, these big red vessels here, those are about 1 millimeter in diameter. And as you're probably aware, blood capillaries can be down to about 5 microns in diameter. So to image all of those vessels all at the same time is quite challenging. So we injected a resin, and it took a long time to sort out the techniques to be able to do this. Injected the resin in this and into these blood vessels and did micro CT imaging to get this image that you're seeing here. So, and then we can use that to estimate the blood flow and the blood pressure, which allows us to say something about the exchange between the lymphatic compartment that surrounds the structure and the blood vessels themselves. And I'm not going to go much into that today, but I do want to show you what we saw just from the imaging is that the surface area, most of the surface area of the blood vessel, of the blood vessels inside the lymph node is distributed around the periphery of the lymph node. Now, these 4 figures are basically just different resolutions in terms of summing up the surface area density. So if you think about, say, let's take one of these big coarse blocks here, that the color in that block would be representative of the total surface area within that block. And if you just pick smaller and smaller blocks, basically you see less surface area. So that's why I'm showing you these 4 different figures. But the consistent conclusion from all 4 figures is that most of the surface area is actually distributed around the periphery. Now this plot, these figures on the right are not from my lab, but they show using fluorescent imaging, at least for some of the larger blood vessels. They're not able to image the capillaries with this technique. But you can see how much this blood vessel network changes within the first couple of days and actually continues to change over the period of a couple of weeks in response to vaccination. So, what happens to the surface area distribution? What happens to the fluid exchange between the blood vessel and the blood vessels in the lymph node compartment? And that's a hugely interesting topic, and we'd love to know more about that. So just a summary of what I've showed you today, lymph node swelling is, our agent-based model at least predicts that it should enhance T cell activation, and Professor Bocharov is right, that this suggests a series of experiments that should be done. And hopefully we can at least do some of those experiments or convince others to do them as well. There are a lot of interesting hypotheses that we get out of that model. Under baseline conditions, most of the flow goes around the low resistance pathway, but with vaccination, that strongly shifts the fluid, incoming fluid, to the paracortex. And I really wish we could investigate lymph node blood vessels more thoroughly. I think that's going to be absolutely crucial to the process. So with that, I'll close up with acknowledgments of the people that helped out this research and look forward to answering questions.
Thank you very much, Professor Moore, for this very interesting and spectacular presentation. And we have time for some questions. Please, as I mentioned before, our listeners can ask questions in the chat or can raise hands. And waiting for their questions, I would like to ask you what you explained, that the flow pattern in the lymph node changes during the infection or as a result of vaccination. So instead of going around, in fact, it starts going directly inside the lymph node. So could you please explain or maybe give some suggestions why it happens? What is the mechanism which changes the flow pattern?
We don't know. It could be that the connection between the subcapsular sinus and the medullary sinus. Well, this is our best guess. We think there are some cells at the connection between those two compartments that contract in the presence of some antigen, and therefore shut down that low resistance pathway. So you don't necessarily get a greater pressure difference across the lymph node because the vessels that do the pumping, they can only generate a few centimeters of water of pressure. So if you want to shunt fluid to a different part of the lymph node, basically you can change the relative flow resistance of different parts of the lymph node. And I think that's what these cells are doing. But like I said, there would not have to be very many of these cells. So if you do flow cytometry of every cell type you get out of the lymph node, first of all, you'll get a lot of different types of cells, and very big numbers of T cells, obviously, very big numbers of B cells and other cell types for FRCs, and then how do you find, it's like a needle in the haystack, how do you find these few cells that are the flow controllers? And that's not something we've been able to do yet, but we hope to soon.
Okay, thank you very much. And Gennady Bocharov raised a hand, so please, Christina, could you allow Gennady to ask a question? Yes, please, Gennady, go ahead.
Thank you very much. It was a great talk and a great, study. I'm very impressed. Congratulations. My question is about the, following. In your, agent-based model, did you see the results of the flow patterns of the lymph flow patterns analysis in the cortex area to consider the distribution of the chemokines that should guide the movement of the cells? And if and the second related question, is it a 2D or 3D setting?
Yeah, so I'll take the second question first because it's much easier. So it's 3D. So so that agent-based model is full 3D. The first question, no, we did not. Originally when we set out on this agent-based model, we thought, okay, let's spend a few months making a nice agent-based model so that we can couple it to the chemokine transport model that we already had. And then the few months of making an agent-based model turned into a few years because, as you correctly pointed out, it's quite a complex system. And to get it, to get all of the interactions captured is very difficult, and then to fit that to the experimental data, as you alluded to, as well, is very difficult. So it took us a long time to get that agent-based model to the point where we believe it, and therefore try and get others to believe it. And but I will say, we did, as you did, based our parameter estimation on as many experiments as we could find. And then actually we validated the performance of the model by comparing the outcomes to other experiments that were not used to make the model. So, we did our best to validate it, but it's such a complex process. But we do hope to do that coupling process soon.
Thank you. Just a small, just a quick, just a short question. For collaboration of the cell migration, like histograms of speed distribution, meandering, the turning angle. Did you see some regional data? Did you see those that were presented by, for example, John Timmis in his work?
So our T cells exhibit a random walk behavior, so roughly linear with square root of time. So and I think that's appropriate in this type of model. I think once we couple it to the chemokine transport model, then you'll start to see more directed movements and maybe things like subdiffusion or superdiffusion. But it's not in there yet.
Okay, thank you very much.
Thank you.
Thank you.
We have one more question from Alexei Tokarev. Please, Alexei, you go ahead.
Yes, thank you, Vitaly. Mr. Moore, I have a question. In your experiment, you talked about the local lymph node, which is close to the area of injection. But does it swelling occur locally in this very local lymph node, or it is body-wise? How do you see? What do you see? You see at other lymph nodes too, and what is the body-wise mechanism?
Yeah, it's a very good point. We did not look at other lymph nodes in these experiments. I think that's something that would be very interesting to do. In particular, you know, in the mouse, there is an unusual connection between the lymph nodes in there's a lymphatic vessel that kind of runs up the side of the mouse, which we don't have in humans, but it makes for very interesting structures and interesting communication between the lymph nodes. But it's yeah, there certainly will be system-wide reactions, and I suspect if we were to look at the other lymph nodes in the mouse, that they would also show some signs of swelling. But you know, that's a good topic for a future project. So maybe you or someone else can take it on.
Okay, thank you.
Thank you for the question.
Okay, thank you very much for this discussion, and again, Professor Moore, for this.
My pleasure. Thank you for the invitation to speak.
Yeah, it's a pleasure for us. Thank you again, and we will move to our next speaker. Anas, please, you can share your screen.
Thank you, Vitaly. So first of all, I would like to thank the organizers of this workshop for the invitation, and let me look for the slides. So I hope that you are able to see the slides.
Yes, we see. Sorry, we see the slides, and I will use this occasion to shortly present you and your presentation. So our next speaker, Anas Bouchnita from the University of Texas, will present us seroprevalence modulates key epidemiological characteristics in COVID-19. Please, Anas, we are listening to you.
Thank you for this introduction. So first of all, I would like to acknowledge my colleagues at the University of Texas at Austin and also at Hong Kong University for this joint work. And I would like to follow this outline. So I will begin with an introduction of what is seroprevalence and what is our knowledge of its effect on the transmission dynamics of COVID-19. I would like to then introduce a multi-scale within-host between-host model of SARS-CoV-2 and show some numerical simulations. Then I will derive a novel, simple model that falls into the SARS category, and that considers the effect of seroprevalence explicitly on COVID-19 transmission. So, as you know, all of us have heard of this concept of herd immunity since the beginning of the ongoing pandemic. So, it represents the situation where there are a high level of immunity at the population, and where there are individuals that we can show here in this screen that will form a sort of a shield between around an infected individual and prevent them from reaching this susceptible population. And there is this concept of herd immunity threshold that postulates that there is a certain level of the population that should be immunized in order for the pandemic to start disappearing or to start going down. So today our understanding of COVID-19 have changed in this previous year. So even though we've seen the many successful vaccination campaigns in countries around the world, we still have new waves of infections every now and then and at different regions of the globe. And so this promise of reaching this herd immunity is fading away, and there are different explanations for that. So first of all, we have the possibility of reinfections that was demonstrated. So individuals who got the COVID in previous year, they got it this year too. We've also seen the emergence of new variants of concern, and these are these mutations that confer some new features to the virus, like becoming more transmissible and causing more severe disease. And we've also had evidence of the waning of acquired immunity. So now we are talking about booster shots, and the argument is that this acquired immunity starts waning after a certain number of months. So now the question is whether the models that we've been using at the beginning of the pandemic and which use these estimates for these epidemiological characteristics, like the rate of symptomatic individuals, the rate of hospitalization, the characteristic times of the different progression compartments, whether they remain the same or whether they change when this immunity build up in the population. So here we use the term seroprevalence, which means the prevalence of antibodies that confer immunity against the disease into the population. And so to investigate this question, we have devised a new multi-scale immunoepidemiological model, which means that it's a model that couples viral kinetics, individual within host viral kinetics, with an agent-based framework for infection transmission at the population level. And so at the within host level, we will use a limited cell model that describes the interaction between the viral infection and the infected cells with both the innate and the adaptive immune response. And at the population level, we would use what we call a social force model. So these are agent-based models that simulate the motion of pedestrians in different settings, like in crowds, under panic, and so on. And we will use a probability of transmission that depends on the viral load of infectious individuals. So this is what the model will look like. So this is an agent-based social force model, and inside of each of infected individuals, we will solve this within host model, which will be a simple ordinary differential equations to track the evolution of the disease and also the infectiousness of infected individual. So let's start with the within host model. So we use this limited cell model. So here we have infected cells, healthy cells, the viral load, interferon, which participates in the innate immune response, and the antigen-specific T cells and B cells, and the antibodies. And one important feature of the model is that we solve this system during infection, but as soon as the infection is resolved, we keep tracking the number of antigen-specific B cells and T cells and also antibodies inside of the individuals to simulate their level of immunity. And we use some estimates from the literature and from the data that give the half-life time of antibodies and antigen-specific T cells and B cells following infection. And so one of the challenges of these models is how to simulate the outcome of the disease or the evolution of the disease. And of course, there are different indicators that were used in the literature, but one of the most widely used one is the area under the curve of the viral load, and it estimates the amplitude of the infection. So when an individual has a high viral load, then the area under the curve will be higher, and this will impact the different systems and organs, and will probably cause hospitalization and also death if this concentration is very high. And so we've seen this applied to models that study influenza, but also to some recent models for COVID-19. Another challenge that was also raised in the previous talk is how to estimate the patient-specific parameters and what parameters depend on individual patients and which parameters do not. Of course, it's a very complex question, and if we change all parameters, then unless we use some nonlinear regression model to make the estimation. It's very complex. But there are some differences or some parameters that we know that they depend on the features of individuals. For example, we know that the age of patients or of infected individuals determines the level of naive lymphocytes in their organism, and this gives us an idea about the strength of the adaptive immune response. So we consider that these parameters depend on the concentration of naive T cells and B cells depend on the age of individual. And another parameter that we change is the threshold of the area under the curve that can cause the progression of the disease. So we know that people who belong to high-risk group are vulnerable, and therefore small concentration of the virus could cause serious outcomes. So the next step is to validate the model, and to achieve this, we use both the estimates and the literature. And for the remaining parameters, we use the fitting of the viral curve with the available measurements for the SARS-CoV-2 viral load in swab and sputum also in the literature. And we also make some comparison with the level of antibodies following infection. And for this, we use different levels of half-life time to make robust analysis. But we still don't know whether the model works very well at the population level. And so to answer this question, we generate a cohort of virtual patients that consists of 1,500 patients and who have their age and risk group sampled according to the demographic data. And so we assume that all of them get infected, and we see how they would respond to the disease. And by fitting this area under the curve, we could reproduce the epidemiological characteristics of COVID, or at least the wild type Wuhan version of SARS-CoV-2. Next, we have studied the effect of aging on the response or the viral load of individuals upon infection. And to do this, we changed the levels of naive lymphocytes. And we, as expected, we've seen that the younger individuals will be able to contain the infection very fast. While people who have low number of lymphocytes will have a persistent infection and will have severe disease outcome. At the same time, we have also studied the effects of the initial level of memory lymphocytes and antibodies on the progress of the infection, and we have seen that having only 20% of antibodies is sufficient to prevent hospitalization and death. So the next part of this is to incorporate this within host model into the agent-based framework model. And as I said, we will be using a social force model. And this model uses the Newton's second law of dynamics to track the motion of individuals. And so we approximate individuals by particles that are subject to three forces: a social force, a self-driven force, and a wall force exerted by the wall, which here will be considered zero. And instead, we will be considering periodic boundary conditions to approximate larger systems. And what remains is estimating the infectiousness of individuals. So when two individuals get into contact, which means that the distance between them become very close for a longer period, then we estimate this probability of disease transmission, which depends on the viral load of the infectious individual at this period, which means that if an individual has a high viral load, which usually happen during the day of the incubation period or the day before, then they will be highly transmissible and they will have a higher chance to contaminate the individuals upon contact. And so we ran some numerical simulations. So we consider a population of 500 individuals. We began by introducing 3 infected, or considering that 3 of them are infected at the beginning, at the latent stage, and we follow the progress of the infection. We've seen that there is some sort of a peak that happens in month 2, and after that we have a stable number of symptomatic individuals and hospitalized individuals. So the model does not predict that the infection will end after this first peak, which is really interesting and realistic. So, these are estimates for the seroprevalence. So here we see the levels of antibodies in the different populations. So during the peak, we see that there are few individuals that have a higher level of antibodies, but a few times after that, then there are a larger group of people who have antibodies, but their level of antibodies starts decreasing. And here we can see this dynamics in these histograms. And we also have this population dynamics results that show that there is a first peak followed by a sort of stability. And we also see that the number of cumulative cases and number of cumulative infections are somehow the same, but there are some infections that would not lead to, or there are some infections that would not lead to disease or to symptomatic disease because most of the population will have these antibodies. And so we have also some other results like the time of infections, which increases following the age and also the time of infection. But the main results that we have observed, and I think which answers the question that's of this whole talk, is that as seroprevalence starts to build up in the population, then the rate of infection decreases, the rate of symptomatic decreases, the rate of hospitalization decreases, and the number of deaths also decreases. So we have started to think about a way to incorporate these dynamics into simple models in order to gain insights into the evolution of the disease. And so we know that most of the existing SIR and SEIR models, epidemiological models, they divide uninfected individuals into 2 groups, susceptible and recovered. But in COVID-19, susceptibility is relative, which means that there is no such thing as being removed. So you always have a chance of getting infected, and there are always cases of reinfections. And but these reinfections are most likely would result in asymptomatic disease or less severe outcomes. And as I said previously, so seroprevalence reduces the rates of infection, symptomatic disease, hospitalization, and death. So we've come up with this cheap model to track the level of or the effects of seroprevalence on the evolution of the disease. So the model is a compartmental model of the type SEIRS. So with reinfections, and we introduce this variable called here M, which means the seroprevalence, and this variable affects the transition rates between the groups, and it increases when there are these recovered individuals. So it really represents the effect of seroprevalence on the dynamics, or at least this is what we could observe in the multi-scale within between hosts model. So we did some fitting of the model, the ODE, the simple ODE model, to reproduce the results of the multi-scale immunoepidemiological model, and we were able to obtain a nice fit, except maybe for the infection for the fatality ratio, which was not very representative, but the model is able to reproduce this dynamics, and we observed several interesting results. So first of all, as long as you have the waning of the immunity, then it will be impossible to reach herd immunity. So as long as you have even small levels of herd immunity, then it's impossible to end the pandemic, because there will be always some leaks that will fuel the pandemic. And as you can see here, these are simulations for years, but with without immune waning. And in this particular case, it is possible to have herd immunity and to speak about the threshold of herd immunity. So if you have a certain level of immunized individuals, then they will lead to the disappearance of the pandemic, if we can say that. And next, we introduce this immune waning, and we show that in this particular situation, we will not have the complete eradication of the virus, but instead we will have an endemic state, which means that the virus will become less lethal. It will keep circulating, but it will become much less lethal and will provoke much less hospitalization. And also the infection fatality ratio will be very low and will reach the IFR of the influenza. So the model allows us to make this long-term prediction of many years. So here, for example, we've seen the effects of ongoing vaccination campaigns. So when we start giving these booster shots every 6 months or whatsoever, and we see that it's a possible way of controlling the number of infections. It will not eradicate the virus. The virus will always keep circulating, but it has the potential to save lives. On the other hand, if we give the vaccination for a short period of time, like 6 months or 3 months, then as soon as we stop giving this vaccination, then the number of infected individuals and also the number of hospitalized individuals will converge once again to the initial solution. So one last result is the basic reproduction number. So we were able to estimate the basic reproduction number for this model using the next generation method. Interestingly, so when we consider high contact rate, which is now the case with this variant of concern, so the contact rate, the corresponding contact rate for the Delta variant is about 0.005. So in this particular situation, it will be impossible to have a basic reproduction number that falls below 1, which means that it's less likely that we will be able to control the pandemic and eradicate the virus. So there will be always an increase in the number of cases. But as I said, the model shows that most of these cases will be not, or at least the disease will be less lethal. So here we see the effects of seroprevalence, or the fraction of immunized population, on this basic reproduction number for different transmission rates. And if we consider higher transmission rate, like in this purple curve, then it will be always above 1. And also, it is very hard to keep the seroprevalence at very high levels, now that we know that it's impossible to, or that the immunity is waning. So to conclude, we have shown that seroprevalence decreases the chances of disease transmission, symptomatic disease, hospitalization, and death. And I think this is a feature that should be incorporated and studied using existing epidemiological models, because it's very realistic. The model shows that if we have this variants of concern like the Delta variant, and now we also talk about the Delta plus variants, then it's less likely that it's more likely that herd immunity will be impossible to reach. So now we have 2 tools. We have to keep the seroprevalence high, maybe using booster shots and natural infections. And also we have to keep the contact rates very low using, or the transmission rates very low using face masks and physical distancing and so on. And hopefully this will morph the COVID-19 into a less lethal endemic disease. Thank you very much. That was everything for my talk.
Thank you very much, Anas, for this very interesting presentation, and you showed several new models and really questions to discuss. So we have some time to ask questions, and I recall our listeners that they can also ask questions through chat or through or raising hand. And waiting for their questions, I'd like to ask, first of all, in the first part of your talk where you presented this agent-based model. By the way, this immunoepidemiological model, this is really very interesting because we are thinking about that in another framework more related to continuous models, particular ordinary differential equations, but it's quite difficult to bring together these two parts in a more or less realistic model. So this agent-based model, it's really something interesting from this point of view. So when you showed the motion of individuals in your model, you mentioned several forces, in fact, 3 forces, and among them social force and cell force. Could you explain, please, what you understand by these forces and how you define them?
Yes, thank you, Vitaly, for this interesting question. So the model assumes that each individual is subject to 3 forces. So the first one is a self-driven force, which describes the tendency of individuals to go in a certain direction and to keep a certain distance or a certain velocity, which is called the desired velocity. And the second force is the social force, and this force describes the tendency of individuals to keep a certain distance between them and other individuals, and can depend on different settings. For example, we know that individuals tend to keep smaller distance when they are at home than if they are at work or in a public area. So it depends on this this desired distance, and also an amplitude which describe the compliance of individuals to like physical distancing or their willingness to keep this distance. So this is a framework called social force model. It's very well studied, and there are lots of works and a lot of versions and improvements for this type of modeling.
Well, unfortunately, truly speaking, I'm not familiar with this work. So, for example, if you want to take into account like people going like to their works and gathering together, for example, or children going to school, so for some time during the day. So, how do you take into account? Do you take this into account in your model?
Yes, this is a very good question. So one way to do it is to consider like this realistic geography, where you consider like, for example, a neighborhood or an avenue with different households and workplaces and schools and so on. And then you can change the schedule of people going to these different areas. For example, for people going to school, they could have different self-driven force that would push them to go to school, and then as soon as they reach this place, then they will keep circulating in this particular region. So there are ways to introduce this, but for this particular work, we have considered like this that people can go in random places, and we don't have this heterogeneous pattern dynamics to not make the model more complicated or the results more complicated to interpret. But we have also some ongoing collaborations where we study different particular settings, like, for example, the corridor settings, where there are 2 groups coming from 2 sides, and we can study the transmission risk in this particular settings.
Yeah, thank you very much for this answer. And since we don't have yet other questions, I will continue with my questions because I have many of them. Another point which is very intriguing for me in this epidemiological models, or maybe it is better to say not in the models, but in the data. I will begin with the data, and then I will ask a question about your model. So we observed in the data for example, annual influenza epidemic and for other epidemics, that in fact we can observe what we call over exponential growth of the number of infected individuals. So I will explain what I mean. If we take just the usual case of, for example, SIR models and things like that, so in the beginning, the number of infected individuals grows exponentially. But considering some data, we observe that this can be different, in fact, that in the beginning of epidemic, there is some exponential growth, but after some time, when the epidemic limit is overpassed, then there is an acceleration of growth. It will be also exponential, but with another exponential rate. So this is my point. So there is an acceleration, not just exponential, but two different exponentials. Have you observed this in the data? And what do you think whether these immunoepidemiological models can explain this phenomenon?
Yes, this is a very good question. And I think I've seen work studying this particular question, but they use two types of model. They use this very simple SEIR model and a network model with this heterogeneous contact patterns. And they show that there are some conditions on this for these network models where we could observe this initial exponential growth like in the SEIR model. But in some case, when there is heterogeneous contacts, like heterogeneous contact patterns with different contact matrices and very complex contact patterns, then it's a different dynamics. So I would say that I don't know if it could be possible to study this using this type of model, because we know that infectiousness is sharp at some level. Maybe this would influence this exponential growth, but I think it would be more interesting to introduce accurate contact patterns using, for example, a network model or like a contact structured model, maybe.
Okay, thank you for this answer, because my understanding of this acceleration phenomena, which is, from my point of view, very important for understanding of epidemic, is that when the number of infected individuals becomes large, there is kind of accumulating effect. So simply speaking, you meet not just one infected individual, but you can meet several of them at the same time, and it will increase this infectivity rate. But it's really an open question, and I think it's really very important, and it's not yet studied in the literature, strangely enough, in spite of its importance. And one more question I have, since I don't see other questions from our listeners. You mentioned that for this Delta variant, it is unlikely to reach collective immunity. I'm a little bit surprised by this conclusion. I understand that the infectivity rate is higher and the basic reproduction number is also higher. But still, if the vaccination rate is high enough, we can expect that we will finally stop the epidemic. So please be optimistic. Tell us that we still have a hope to finish this epidemic.
Yes, I would love to say like optimistic word about the end of the pandemic, but I think that now the focus should be to have this transition to our endemicity and to our COVID-19 becoming less lethal. Of course, the model that that give us this conclusion is quite simple. But we have also seen this in other models with reinfections and immune waning. So we keep having these new waves. And of course, there are different solutions depending on the parameters. But I would say that as long as seroprevalence is high, we should have COVID-19 becoming less dangerous and less lethal, hopefully.
Yes, of course, I agree with you that once recovered, become again susceptible after some time because of the decrease of the level of antibodies, the same with vaccinated. So there will be a constant influx of susceptible individuals. And then, as you say, we'll have this chronic infection with this endemic equilibrium. And then the level of this endemic equilibrium whether it is high or low. It depends, of course, on the intensity of vaccination and on other factors. Okay, thank you very much again for this really very interesting and very enriching presentation with all these new models. And then we can gradually prepare our next presentation by Rostislav Savinkov. We have several minutes. Rostislav, please, you can share your screen, and then we can wait a couple of minutes before the beginning. Yes, we see your screen. So let's make a short break, 2 minutes, to respect our schedule, and then we will start the presentation. Okay, we are back from our short break. Rostislav, do you hear me? Are you here?
Yeah, yes, I hear.
Okay, then we will start our next presentation. Rostislav Savinkov is co-author, and Rostislav will tell us about modeling individual cellular dynamics using cellular automata and artificial neural networks. Please, Rostislav, you can start your presentation.
Thank you very much for the introduction. I'd like to tell you today a few words about computational algorithms for the modeling of the cellular automaton methods. And there are two main tasks in my work. The first one is the development of computationally simple methods for calculating the deformation of the cell body. And the second one is the determination of patterns when calculating the time of cell migration in the field of stackings. The second one could not be solved without the first one for the large amounts of cells. Let me explain the first problem. First problem is the difficulty of the cellular deformation while modeling, because we should every moment of the model check all the cells to keep the bodies as a solid structure, and to check the surface to be the monosurface, to exclude the internal voids inside the cellular body. And the difficulty of these processes depends on the volume of the cell and the surface square of the cell. The first one check is very difficult, so it was considered to be necessary to find the way to model without the full cell complexity checking. And we worked in this way for now just for two-dimensional representation, but in the nearest future we expect to go to the three-dimensional version and finally get good mechanisms to model the massive groups of cells in the adequate time, not to wait for days or weeks or months to model some about maybe 1 million cells, maybe. While working, we found some set of patterns for two-dimensional variant. You can see here the growth patterns. There are the 42, if I'm not mistaken, templates, and you can see on the right side of the slide the examples of using these patterns. So if we have some cell, I'm sorry, do you see my pointer?
Yes, yes, we see your pointer. Yes, please, you can continue.
Well, so we have some cell in the computational area, and we can check the boundary of cell for the some template of the presented. And if we find the following template in this list in the left side, so we can add a new segment for the cell body, and using these templates guarantee us the two following rules. The voids would not appear inside of cell body, and the cell body would not be broken. And later we'll explain more. Also, cell can't just grow for the whole time, it should reduce. Reduction we can execute with the following set of patterns, and using these patterns we also provide the required properties of deformation. As a result, we can just check the boundary of the cell for each deformation, and we shouldn't have to check the whole body, and shouldn't compute the task with complexity n cubed, where the n is the linear size of the cell. After that, we should define the direction of cell movement. There are various schemes for that. We can use just a vector to the maximum concentration of the some hemokine that we use in the model, or we can make some distribution and calculate the possibility of direction, and one of these directions by the by its possibility, or we can use random direction, and it would be some random walk for the cell. There are a lot of different other variants, but no, well, you can offer your own. It's not matter. The main target of these schemes of direction selection is to make the model to act like a some real data, if we have it. So using these algorithms, we realize the model and compute many realizations of the cell movement inside of some different types of computational area. Here you can see the two interesting types. The first one is a Petri dish environment, where we can see a lot of similar objects that would not be moved, and the cell should move between them to achieve the target point. And the second one is the two-dimensional layer of the three-dimensional model of the fibroblastic reticular cells network. And here we also made computations for the 1000 cells. And we placed randomly the target point at each experiment and the starting point of the cell. Also, we executed the simplest variant where we have a free area without any objects, just a target point and the cell. And here you can see the dependency graphic of the dependency of the time contact from the initial distance between target and cell. We created the linear regression of this data, and you can see this. It's rather good. Okay, it's a free area. It should be so. After that, we created the very simple neural network that should predict this dependency based on this data. For free area, it is very similar with linear regression. But later we will see the large differences for other areas. For example, the Petri dish. We had a computational area size of 400 micrometers, the linear size, and 640 dots of some fixed size also. So in this area, we put target point, put the cell, and compute the time of contact. It is rather interesting that in this area we have less time to contact than with free area. I don't know why, but it is very interesting. After that, we created the linear regression. Okay, it's rather good in the middle zone, but on the ends it's not correct. And using the same neural network, we get this line. It's much better, and Here we can see the good result of modeling of the curve. But on the next experiment, we will have not very good results. I will try to explain why. In the fibroblastic reticular cells area computation, the same rules, the same cell, the same algorithms. By the word, we used the stochastic direction vector distribution for the cell while modeling. I told about it before. And here we get such strange data. We have large outcomes from the main line of the contact time. And in this, with this data, we have some linear regression, and we have strange artificial neural network approximation of the contact time. In my opinion, it depends on these outcomes, because in the main path looks like this. Some curve that goes up, but the artificial neural network decided that the best curve that approximates the presented data is such a curve that we see. Maybe it would be better to use the logarithmic values of the time contact to model in the further time. But for now, we have this. As a result, first of all, for two-dimensional case, we developed and test the algorithm that allows us to model the massive amount of cells in the same time, or model one large cell. Large, I mean consisting of thousands, maybe, or hundreds of thousands dots in the computational area. On the second step, we could show on the free area and on the Petri area that the even the very simple artificial neural network can generalize significant information that we can get from this model that allows us to predict the time that cell needs to contact the target object using just the chemotaxis and without some additional computations, just with simple checks of the local states. I think that's all. Thank you for attention.
Okay, thank you, Rostislav, for this presentation. We have now some time for discussion. Please, if our listeners have questions, they can ask. And waiting for other questions, I will begin with some questions to clarify, to try to clarify for myself what you explained. For example, in the beginning, you showed some rules how cells can grow or in size or decrease their size. Now, the previous one with this, I think the previous slide. Please, could you show the, yes, this one. Yes, this one. You explained with this square, well, network. You put it on the cell, and like that you determine whether the cell will grow, for example. So what are the rules exactly? I'm not sure to understand precisely. What are the rules which determine here that the cell can grow?
Here is the reduction patterns. They are not the. I'm sorry. These patterns led the cell to decrease the size of the body because for the cell that has some volume, okay, we repeat the step of growth, the step of reduction, and every step we can grow somewhere one segment and reduce somewhere one segment, and with these actions, repeating them, cell gets an opportunity to move through the computational area. If you let me, I will show some small video.
Yes, please. You have time. You can explain indeed with some videos if it is not difficult for you to show.
Is it visible?
Yes, we see your screen. Yes.
Okay. Here the green point is a cell in initial position, and the red point is the target. There is some diffusion in this area, and
so what it is? It's chemotaxis here or what? Because this red dot, what you call target, and there are some. It's some concentration of something around it. What is this? Just
yes, here the concentration of the chemotaxis is presented by the red, but it's not visible in the main area, just around the target point. But in fact, the chemotaxis is working all over the computational area, but I'm sorry, it's just problem for visualization.
Okay, so your green cell moves just the gradient of some molecule, and it moves along the gradient, going around these obstacles, right?
Yes.
Okay, so now it's more clear. And then when you showed these graphs, how the contact time depends on the initial distance, please return to the first graph. The first graph was linear. Yes, this one. Yes, this one. So this is also in the case of chemotaxis, right?
Yes, yes.
Yeah, this is what was not clear for me, because I thought that this was for a random motion, and for random motion it should not be like that. Okay, so but in the case of chemotaxis, indeed and by the way, the cell will speed how fast it moves. Does it depend on the concentration of these molecules, or just its speed is constant?
In fact, the linear speed is constant on the large time of modeling, but locally it could change because of stochastic algorithms of cellular deformation.
Okay, but in average it is constant. I mean, in your model you don't impose the dependence of cell speed, for example, on this concentration of this chemotaxic molecules, no?
No, the
Okay.
concentration of molecules doesn't.
Okay, so this is why you have this linear dependence, because in fact, in average speed is constant. But then, so this is for the case without other cells, as far as I understand, right? In the empty space. But then you consider the case when you have many cells. I think it's your next figure. Show, please. This one. Here you have many other cells, right? And
some static cells. Maybe.
Sorry?
No, yes, we can. We model the static objects.
Okay. You have many fixed cells, and you have one cell which moves to the target, yes?
Yes.
And the graph is a little bit different. So in fact, the dependent is slower than linear, right?
Yes.
So how can we understand that? So if you increase twice the initial distance, the time will not increase twice, but it will increase less than twice. So can we understand it from the physical point of view, why in fact this is like that?
Maybe it depends on the algorithm of target selection. In our model, we selected it relatively to the hemokinase concentration, and in the free area zone, maybe there are more ways to move not direct to the target, but to the some side way.
But here you have only one cell and one target. I mean, one moving cell and one target. Yes.
Yes.
Okay. Okay. And finally, the next numerical experiment you show this the network of this fibroblast, right? Yes, this one. And again, the cell moves to the target, and again, this dependence is different, right?
Yes.
So there is some tendency to increase. So the speed, this contact time looks like it increase more than the distance. Okay, so yes. Okay, so well, of course, these are very interesting results. Sorry, I have some telephone call. But yes, indeed, it would be really interesting to give some physical explanation about this dependence, how this contact time depends on.
I'm not sure is it significant to look at the fibroblastic reticular rod cells modeling in this case, because here we have the two-dimensional organ, in fact, but we should look at the three-dimensional organ. And there we will have very different.
Well, I agree that maybe in 3D it can be indeed different compared in 2D because there will be different mechanism how cells can go around the obstacle. Yes. Okay. And so you are going to apply probably this method to study probably the lymph node, or what is the just further objectives of this work?
I'm sorry, what?
You are going to apply this model probably to study the immune response. So could you please, since we have some more time, could you please explain how, to what particular application you will use this model?
Yes, in fact, well, when I will complete the three-dimensional variant, we will try to apply these methods to model the immune response inside the lymph nodes. For example, while HIV infection, there are about from 18 to 24 hours when the infected cells are still not producing the virions, new virions. And if the affected cells can reach the infected cell in this time, it makes possible to stop the infection at the early times, and it would not produce the viral proliferation. So it's significant to see how much effector cells are necessary in the organism to stop the infection with some potential effectivity.
Yes, indeed, it's a very interesting and important application. But what you say is that infected cell, it takes some time before new viruses will appear, will be produced in these infected cells. So there is some time delay. But as far as I understand what you say, even before that, this cell can be already recognized by cytotoxic T lymphocytes, right? Even before it starts to produce new viruses, it can be recognized probably due to some chemokines
Yes,
or other molecules. Right?
cell starts to produce some chemokines that announce the effect of infection before starting to produce the really effective virions.
Okay, thank you very much. It was very interesting. If there are no other questions, then we'll have another break about 8 minutes, and then we will return after this break to our last presentation of this section. So we meet in several minutes. Thank you again.
Thank you very much.
Well, we are back to our session. And Christina, do you hear me? Are you here?
Yes, I am here.
Okay, so our next speaker, Christina Leon. By the way, I would like to say that Christina among the organizers of this workshop, and she was very helpful in preparing. And even she, at this even moment of time, she is a technical moderator also of the session, not only speaker. So Christina will tell us about. Sorry?
Only one person.
Yes. So Christina will tell us about modeling of the innate and adaptive immune response to SARS viral infection. So please, Christina, you can start your presentation.
Thank you. Good morning, everyone, and welcome to my presentation. First of all, let me thank you all for coming here today. As you can see on the screen, our topic today is modeling of the innate and adaptive immune response to SARS viral infection. To get started, I would like to give you an overview of process of viral infection by virus of coronavirus type. On the right side, we can see a 3D model of SARS-CoV-2 variant. The small protruding red heads are the spike proteins, thanks to which the virus enters to the host cells to begin its replication. With the presence of the antigen in the organism, the immune response begins, which develops in two stages: innate immune response, the first defense line, and adaptive immune response, responsible for the production of specific antibodies for virus neutralization. With the arrival of SARS-CoV-2, many questions arose. The answer to this will help us better understand the virus and thus develop a strategy for how to combat it. In search of answer, through our models, we set out to understand the causes of high interpatient variations in symptoms of the severity of the disease, cause of long incubation time followed by severe or mild manifestation of the disease, abnormal inflammation response, that is cytokine storm, in the part of patients, and also establish the values of the kinetic constants, many of which are unknown. For the development of the models that we will see today, we have used general models of the immune response in general. We have calibrated our models, combining them with the experimental and clinical data on SARS-CoV and SARS-CoV-2 infections. The models are validated by the agreement between the results and the experimental data. When the virus enters the host, it begins to spread in the host by infected epithelial cells. The second green box represents the antigen-presenting cells, such as dendritic cells and macrophages. The infected antigen-presenting cells and infected epithelial cells stimulate the secretion of cytokines, such as interferon. This modulates the function of the immune system and inducing antiviral state. I would like to emphasize that the innate immune system is not reduced to this scheme, since many more cytokines and chemokines are involved in this process. However, considering the information studied, it is precisely the interferon that plays the main role in this stage when we tell about respiratory viruses. Why is this model specific to SARS viruses? Because other viruses of the coronavirus type, such as MERS, for example, can also replicate in infected antigen-presenting cells, while the SARS-CoV and SARS-CoV-2 viruses infect them abortively. This virus is not replicated in them. The adaptive immune response stage begins with the recognition and distinction of non-self and self pathogens. The antigen-presenting cells carry on their surface an MHC molecule, through which they present fragments on the antigen peptides. This antigen presentation process stimulates the activation of T lymphocytes, and this process to differentiate mainly in cytotoxic T cells, or commonly named T killers, and T helper cells. T killers are responsible for destroying infected cells by the antigen. On the other hand, T helper cells, when contacting naive B lymphocytes, activate them and stimulate their proliferation. Activated B lymphocytes are also known as plasma cells that will be responsible to produce virus-specific immunoglobulin, or commonly known as antibodies. To begin the analysis of this model, we concentrate, for now, in the innate immune response. The main objective in this part is to understand if interferon can suppress the infection at the stage of the innate immune response. This subsystem consists of 6 ordinary differential equations. Here, E0 denotes the initial number of uninfected epithelial cells. The expression K2EV indicates the infection of E cells. In the second equation, infected epithelial cells are generated by the term K2EV, while the death rate of these cells sigma 1. The death rate allows their elimination. Here, analogously to the first equation, we have an initial number of antigen-presenting cells, C0, that are infected by the virus, and they become part of the fourth equation. The parameter sigma 2 symbolizes the elimination of infected cells, CV, either by longevity or by programmed cells death systems such as apoptosis. This equation characterizes the virus population dynamics during infection. The first member of the equation describes the increased concentration of the virus in the host due to its replication in infected epithelial cells, EV, with production rate F0. However, this production of viral copies is suppressed by the presence of interferon. The interferon kinetics is described in less equation, where the secretion of interferon is stimulated by the infected epithelial cells, kappa EV, and more important, by the infected antigen-presenting cells, CB. G0 is the interferon production rate, while G1 is the rate of virus influence in interferon production. Finally, sigma 3 and 4 are the death rate of virus and interferon respectively. From described equations, we can get the stationary solutions. Each parameter of the model plays a significant role in the genetics of the system. So certain changes in the parameter values allows us to observe different interesting cases, such as monostability, bistability, and even periodicity. Monostability is characterized by a single stationary point that has a high value. Bistability is denoted by the presence of 3 stationary points, of which, as expected, the first and third are stable. The periodicity of the system is manifested when the stationary point is not large enough. In this slide, we can see how, in the case of monostability, the virus converges to a single stationary point regardless of the initial viral load, small viral load or big viral load, converges to the same point. In the third figure, we observe how the system adopts an oscillatory regime due to the single stationary point is not large enough. In the middle figure, we set the bistability of the system and how the convergence of the system to one or another stationary point is subject to the value of the initial viral load. Small viral loads converge to the first stationary point, while high viral loads converge to the third stationary point. Simulations of the bistability case allow us to clearly appreciate the behavior of interferon. For low viral loads, after the infection at time 0, the virus begins its reproduction in the organism, reaching its peak approximately in the first day. The presence of the virus stimulates, through the infected cells, the production of interferon that briefly reaches high concentration values and stabilizes in this. On the timeline, the moment of maximum interferon concentration corresponds to the inflection point on the virus picture. After which, the virus concentration tends to its stable value. The lower viral load, the faster the virus converges to its stability value without any considerable concentration of virus in the host. On the other hand, with high viral loads, they suppress interferon secretion, and in such a way, he can reach high concentration peaks of viruses. In other words, the containment of the virus in the first stage of the immune response depends on the viral load. For small viral loads, we can contain the virus in the first stage. For high viral loads, initial viral loads, the virus reaching a peak. Moreover, the group of high viral loads makes the system adopt a more expressive behavior. Within this group, depending on the viral load, an incubation period can be observed. For comparisons, we vary the interferon secretion rate, G0, and we observe periods for incubation and maximal viral loads depending on the initial viral load. Here, the vertical lines represent the threshold between small and high viral loads. We observe that these thresholds are higher when there is a high rate of interferon secretion, such as 700. and that when the threshold is crossed and the disease develops, for values close the threshold, the incubation period is longer. Here, for example. Similarly, we can note that for values close to the threshold, the maximal concentration of the virus will be lower than for higher values. Disease severity showed depends on the initial viral load. Okay, as I mentioned, the immune response is given in the stage. This picture provided by the World Health Organization reflects how the innate immune response acts from day 0, and from day 6, cellular response begins. The first antibodies are represented by immunoglobulin M, joint 5 from day 8. This is why we have delays in our system. Here, from day 6, the T cells act, while from day 8, the B cells responsible for the secretion of antibodies act. Equation 7 corresponds to the T lymphocytes that differentiate in T4 and T8. T8, or T killer cells, neutralize infected antigen-presenting cells and epithelial cells. So it now appears in these equations. T4 or T helper cells stimulates the proliferation of naive B cells. Now, plasma cells secrete antibodies responsible for neutralizing the virus. These cells decay with their respective parameters sigma. In this slide, we observe the simulation of the complete system. The solid lines correspond to the innate immune response. The dashed lines, here for example, to the effect of the T cells in the model. And finally, the dashed dot lines reveal the influence of antibodies in the dynamics of the model. We can clearly see how the presence of antibodies can make the virus drop even before reaching its maximum viral concentration. Something that will happen if only the innate immune response will be considered. We also observe that the T cells by themselves cannot contain the concentration of the virus and disease, clearly dependent on the antibodies. The antibody's effectiveness to eliminate the virus can be regulated through the antibody production rate K5. On the other hand, if the adaptive immune response is not effective enough, that is, the antibody production coefficient K5 is not large enough, the incubation period can be extended, but the virus will reach a peak. Here, the vertical lines denote the necessary value of the coefficient K5 for the virus to be neutralized. These graphs demonstrate how a higher initial viral load demands a high antibody secretion rate, K5, for containment of infection. Okay, now a bit about the cytokine storm model. In the case of respiratory viruses, the infected epithelial cells and infected antigen-presenting cells stimulate the secretion of interferon as the first line of defense of the organism. Interferon in turn activates the secretion of pro-inflammatory cytokines, such as principally interleukin 6. The same virus presence stimulates the secretion of these pro-inflammatory cytokines. These secreted pro-inflammatory cytokines are responsible for regulating the proliferation of other cells of the immune system, such as dendritic cell, macrophages, and monocytes, which in turn also contribute to the secretion of pro-inflammatory cytokines. A dysfunctional immune response can cause this common modus operandi of innate immune response to incite a cascade reaction that can cause hyperproliferation of cytokines. This super strong reaction of the innate immune response can have a fatal outcome for the host. Our model of the cytokine storm differs from that of the innate immune response by the presence of seventh equation, the characterized pro-inflammatory cytokines. And since these stimulate the production of antigen presenting cells, they also appear in equation 3. In the analysis of this model, the stability of the system can also be observed. In this case, the first stable point refers to a normal course of the infection, while the second stable point characterizes the unbalanced activation of pro-inflammatory cytokines, leading to the cytokine storm. In this simulation, we observe how the peak viral load near day 5 stimulates the excessive production of cytokines, causing this imbalance in the production of cytokines and causing the cytokine storm. In conclusions, innate immune shows the stability and threshold-like response to the initial viral load. Above the threshold, high initial viral load shortens the incubation time and increases maximal viral load. Antibodies play the major role in the fight against the virus. High viral loads leads to a disproportionate increase in pro-inflammatory cytokines. There are still many more things to tell you, but in short, based on what these models have shown, wear masks, and better yet, get vaccinated. Please meet the team involved in this research, our leader Vitaly Volpert, Alexei Tokarev, Anna Bouchnita, and I, Cristina Leon. Thank you for your attention. Do you have questions?
Thank you, Cristina, for the presentation. Please, if there are questions or comments from the audience, you can raise hand. Otherwise, well, I would like shortly to comment, because I will not ask questions, because in fact we discussed all that with Cristina before. Just a couple of short comments, waiting possibly for other questions. So what seems one of the important conclusions here is really how disease progression and disease severity depends on the initial viral load. This is one. I think this is one of the questions we need really to understand, not only from the scientific point of view, but also in our everyday life. So these models show very, very clearly that high initial viral load leads to more severe disease, for example, and small initial viral load. In this case, disease may not develop at all or develop in a weak form. Why it is so important? Because in particular, you can remember that in the beginning of this epidemic, one of the questions just discussed in the public and in medical authorities, whether wearing masks can help against the epidemic. Well, now it's kind of obvious. We wear masks, but in the beginning it was a long discussion which took several months, and I think it's a very big mistake, because one of the opinions was that anyway, mask will not protect us against virus. So in particular, this modeling shows us that it's not the question whether mask will completely protect or not completely protect. It's a question of numbers, it's a question of quantities. Even if masks provide only partial protection against virus, against infection, this is very important, because this decreases initial viral load, and as we see, with the decrease of initial viral load, we decrease disease severity. So this is a very practical conclusion of such kind of studies. Another question which I'd like shortly to comment is discussed more in the scientific literature, and it concerns more cytokine storm. In fact, what is interesting here is that cytokine storm is provoked by the infection, by the viral infection. Then cytokine storm starts, then suppose that infection is suppressed. So there is no more virus in the body. So the question is whether the cytokine storm will also end at this moment, or it will continue. So again, we understand that it is initiated by viral infection, but after its initiation, whether it will stop when infection stops, or it will continue when infection stops. And according to the clinical literature, as far as I know, both cases are possible, and there is no definitive conclusion about how it works. And our modeling shows, in fact, that both are possible, that both cases are possible. It is possible that cytokine storm will finish together with infection, or it will continue even if infection is finished. And in fact, this is related to this bistability Christina mentioned in her presentation, that there are possibly two stable stationary points, two stable equilibria in this cytokine storm, and infection can switch from one equilibria to another one, to another equilibria with high value of pro-inflammatory cytokines. And even when infection is finished, the organism can stay at that high value equilibrium with cytokine storm. So it depends, in fact, on the parameters of the model, as we discussed on individual patient-specific parameters. For some people, cytokine storm can continue even when infection is finished. For some other people, it can stop together with infection. And this is why, of course, this is very important for us to be able to estimate patient-specific parameters. Okay, at this point, I will stop my comments since I don't see other questions. So I would like to thank Christina again for the presentation, and also all speakers and listeners of the session. And I would like to recall that we will come back to our workshop 1 hour later, and we will have another plenary talk by Professor Vasilevsky, and we will have a session on cardiovascular system. So we stop for now. We'll meet 1 hour later, and the connection for the next session will start as in all other sessions, 30 minutes in advance. So please, speakers of this next section, connect in advance. We'll check connection and we'll check share screen option. Okay, thank you for participating, and we'll meet later.
Thank you all.